Imagine the power of a wind turbine harnessing energy from natural forces, converting it into electricity and powering your devices and our modern world. Now shrink that turbine to the nanoscale, and we have challenges. Replace the spinning blades with strands of DNA, and instead of wind, use electrochemical gradients as the driving force. This is the groundbreaking concept brought by researchers of a DNA turbine, a molecular machine operating at the nanoscale of life itself. DNA turbines represent a leap forward in the field of nanotechnology, bridging the gap between biology and engineering. Unlike their protein-based predecessors, which we'll discuss, DNA turbines are programmable, versatile, and built using the fundamental principles of base pairing, Watson-Crick theory. Their ability to transform and harness energy at the nanoscale, at the molecular level, could revolutionize applications in biophysics, medicine, and nanotechnology. These molecular machines work on the same principles as transmembrane proteins like ATP synthase, but they have the added advantage of structural predictability and programmability due to being DNA. By harnessing transmembrane potentials, DNA turbines achieve rotational motion, converting chemical energy into mechanical work that is both efficient and scalable, actually. As we dive into this paper, the future of energy conversion and molecular robotics, where biology meets cutting-edge technology, is at the forefront of what these authors are achieving. Molecular machines, as we know them, have evolved from naturally occurring protein-based mechanisms in cells to sophisticated synthetic systems built from DNA. This shift has expanded the applications of molecular machines. To begin, much of discovery and development occurred in the 1970s, where since then we have developed accurate models of proteins like ATP synthase in our cells. This and other natural protein motors make rotary motions when converting proton gradients into ATP. ATP synthase is the foundation of biological energy conversion as we know it. It is basically a rotary motor that converts proton gradients into ATP. Here we also have kinesin and dynein, motor proteins that have become famous for the way they move across cellular and intracellular microtubules. They're also powered by ATP hydrolysis, thanks to ATP synthase. These molecular machines are critical for moving cellular cargo, mitosis, and other biological processes. They have a 50 to 75% efficiency, which is very effective given the energy gradients of biological systems. It seems like they make the most of the energy at hand. Ribosomes are another familiar protein which translates RNA into proteins. It is a very illustrative example of a protein acting much like a printer or assembler for amino acid chains. As these discoveries were refined, by 1982, Nadine Seaman published an analysis of DNA as a structural material for engineered nanostructures, majorly developing the burgeoning field of nanostructure design. By 1990, Tyrrell published more insights into synthetic protein machines. These early synthetic designs sought to mimic natural protein machines, but they faced severe limitations in programmability, scalability, and even stability outside of biological environments. The first DNA walker, published by Ping et al. In 2004, showcased displacement strand mechanisms for programmed motion in nanomachinery. The paper we are discussing today would not be possible with all this background leading up to Rothman's now famous paper for DNA Origami in 2004, using the same displacement strand mechanisms to allow programmable motion in novel DNA nanostructures. By 2010, strand displacement reactions have progressed enough to allow nanobots to react with their environment, make certain computations, model logic circuits, and even control reaction cascades, leading to where we are today, with Shi Pum and others publishing a paper, a DNA turbine powered by a transmembrane potential across a nanopore. DNA machinery, nanopores. Let's discuss a few things worth understanding. Why DNA? The shift from proteins to DNA in nanotech is a paradigm shift. DNA-based machines are not just biomimetic, but also programmable and versatile. offering opportunities to build systems unattainable by natural proteins. As a molecular material, it has unique advantages over proteins for molecular machine design. Base pairing is precise enough to control DNA structure and function, and assembling strands autonomously from complex and 2D, 3D structures. DNA may incorporate chemical modifications and hybridize with other molecules. We have summarized much of this in a table here comparing proteins and DNA nanostructures. We want to describe in detail the concept of electrochemical potential and use our insight to learn about the nanoforce system of our nanoturbine. The results are applicable to many future technological and biomedical applications, like targeted drug delivery, diagnostics using sensors, energy harvesting, industrial applications, and molecular scale assembly and precision. From chemistry and thermodynamics, the Helmholtz free energy F is defined as the energy E minus Ts, where T is the absolute temperature and S is the thermodynamic constant entropy. Not constant, concept. When accounting for individual particles and chemical species, we can determine the differential form for the Helmholtz free energy, where the new symbol mu is the chemical potential of a chemical species I, and the number of particles of that species is n sub I. The chemical potential for a given ion includes contributions from both the electric potential and the concentration gradients, where mu I equals mu naught I plus kT natural log Ci over C naught plus Q sub I phi. Essentially, the chemical equilibrium acts like an energy store. To showcase how this information can be used to determine chemical equilibrium conditions, let's explore and define the equilibrium concentration in a region, beginning with the ratio of ion concentrations. The concentration for two species, in this case sodium and chlorine across two compartments, 1 and 2, gives these ratios, and these compartments are separated by a membrane. The electrochemical potential has an electric term and a chemical potential term, as expected. At equilibrium, the electrochemical potentials of the same species I across two regions must be equal. That is, mu I compartment 1 equals mu I compartment 2. We substitute our previous expression, and after simplifying, see the relationship of concentration to the electric term, where the change in voltage equals negative kT over Q sub I times the natural log of the concentration ratio. Inversely, the Nernst relation given here describes the equilibrium potential for ions across a membrane. It is notable that the membrane is treated as a thermodynamic system and also acts as a capacitor by maintaining separate charge distributions. We have provided an example of solving for the equilibrium distribution for a system with two different concentrations and the accompanying document linked in the description below. In systems with active transport, like the sodium-potassium pump in our cell membranes, steady-state potentials include contributions from the energy sources like ATP and the Nernst potentials together, making up the steady-state voltage. The Helmholtz free energy describes the thermal energy available for the DNA turbine in this paper. Rotations. At a steady state, the electrochemical torque balances viscous drag, and our energy conversion is the ratio of mechanical work to electrochemical energy input. The Boltzmann distribution provides a statistical framework for us to understand the relative probabilities of particles to occupy states with different energies. For ions across the distribution, the probability to be in a particular state is proportional to e to the negative e over kT. We can relate the ion concentrations generally to the intracellular and extracellular regions across a membrane, where we see the concentrations and the Boltzmann factors across this expression. Once again, given a membrane potential, the voltage determines the energy landscape, and the equilibrium distribution of ions can be found. Since cells have zero charge on the macro scale, we can consider how in our model of cells neutrality is maintained. We have the expression for the various ionic species and the density of electrons, which sum to 0. The DNA turbine of our paper is powered by a transmembrane potential across a nanopore, enabling hydrodynamic flows that are capable of 10 Hz speeds. The turbine wound up having several surprising features according to researchers, including reversibility under the right environmental conditions we'll discuss, and suitability for biological applications in their operational stability and function. In the past, molecular machines mimic and surpass nature's designs. The design of systems that operate in these conditions is a tremendous achievement, combining electrochemical gradients and nanoscale motion. The research developed the turbine to do 10 revolutions per second. It's only 24 nanometers wide and tall, and it looks just like you might think. The researchers explained the design has a twisted turbine due to the number of DNA-based pairs, and by adding and removing pairs in the built-up sequence, it is possible to make turbines of any chirality or even straight turbine blades, which wouldn't be very useful. To show that the turbines generate torque and work in a nanopore, the researchers employed visual tracking techniques. Visually tracking rotations at the single molecule scale still represented many challenges to the team. To measure turbine operation under hydrodynamic load as described, super high resolution microscopy was employed. Since the turbine itself is so small, researchers initially described a 900 nanometer long DNA leash used to lower a turbine into its environment and directed it towards a nanopore using a docking procedure. On one end of a DNA bundle are fluorophores placed by the researchers, which allow continuous tracking of the molecules. Continuous mobility tracking with fluorescence microscopy is a method to snapshot micromotions, and the researchers image the system at 5 millisecond increments. The researchers have sub-diffraction limit localization precision, which means without advanced microscopy, the system itself is more or less invisible to the light we see. The scientists utilize a silicon nitride membrane, which is about 20 nanometers thick, with 50 nanometer pores in the membrane, set using electron beam lithography and reactive ion etching. The pores and surface of the membrane was observable with transmission electron microscopy after it was fabricated. The researchers observed unidirectional rotation when transmembrane charge gradients are present. The scientists actually described a complex procedure preparing the solutions the motors are placed in, using diffusophoresis to insert the turbines in the nanopores, at which time the fluorophores were used to track the rotary motion on one end of a DNA bundle. Being rigid meant it tracked the actual turbine rotation. The researchers showed over 200 rotations for many turbines in a brief observational window, but seeing the publication, many turbines' total angular displacement performance was grouped lower, with some high-performing outliers. The authors attributed outlying high-performance results to be mostly due to heterogeneity with the DNA and silicon nitride membranes, and deformations to the crossbars that affect the drag and observed speed in the DNA attached. Once a voltage is applied between compartments and the 50 millimolar sodium chloride solution the researchers described, the turbines are observed to go under an automatic diffusophoresis reaction, which docks the turbines in nanopores. So it seems this is an energetically favorable configuration when supplied the gradient. We have this picture of the overall shape, laterally, and top-down layers, and experimentally measured counterparts in this big figure from the paper. The evolution of molecular machines reflects the increasing ability of humans to mimic and surpass nature's designs. The latest frontier from the nanoturbine team are demonstrating the potential playing field for biomachinery. Let's briefly consider energetic considerations in the biological context. We associate ATP to be the prototypical energy source, and the reaction that releases this energy is a hydrolysis reaction. ATP hydrolyzes to ADP plus phosphorus plus 19 times kBTR, or approximately 19 times the background thermal energy at room temperature. The idea that the turbine is capable of operating in the gradients that exist, driven in biological systems by ATP, is very exciting since cells essentially allow the turbines to function. Outside of biological context, however, there is still a lot of excitement. We previously briefly mentioned that the rotation direction of the nanoturbines could be adjusted, and this is possible by varying the dilution of solute in the turbine's environment. In a scenario where the sodium chloride buffer was very concentrated compared to before, the directionality was reversed. This was also verified using MD atom simulations, where in these simulations the researchers also reported flowing water can be used to drive the turbines. The angular speed of the vanes even increased at higher flow rates, mimicking the behavior of macro-scale turbines. The simulations matched experimental observations very well, indicating the simulation technique is applicable for nanostructures of this size, which is expected. So, to imagine the power of a wind turbine at the nanoscale, we now have a better picture of a structure that is activated by electric fields, placing itself in a provided nanopore with diffusophoresis. They can create vortices in the surroundings capable of moving other molecules. The DNA turbines could operate from electrophoretic or hydrodynamic flows. The direction of rotation is controlled by the chirality of the design, and even can be reversible when changing environmental conditions. They generated tens of piconewtons of torque. With 25 nanometer tall turbines, they are only about 10 DNA strand widths tall. The researchers improved upon the scaling down of turbines by approximately an order of magnitude compared to previous work, which turns out to also seemingly be a publication by the same principal investigator, Shi. The previous rotors looked like rods that fell into the special nanopores at the center and rotated. Researchers simulated a turbine in a dilute solution. Their all-atom MD simulation did demonstrate rotation in the experimental direction in simulated electric fields with 100 millivolts per nanometer charge. The combination of hydrodynamic and electrophoretic anisotropy ratios determine the direction of rotation. Essentially, the hydrodynamic ratio stays at 0.5, but the electric ratio can range from 0 to 1, and we see that Vx can actually switch directions in the right circumstances. A sign reversal may occur when electrophoretic anisotropy changes due to salt concentration, which is according to continuum theory. Here is some technical information about the exact curvature of the turbines, which could be interesting depending on what you're trying to learn. This large figure determines the overall shape laterally and top down. The experimentally measured counterparts in this figure on the top right are comparable to the mathematical model on the left. In this discussion, we've explained what DNA turbines are, why they are groundbreaking. We covered the physics of energy, chemical potential, and equilibrium across a membrane. We described some challenges in creating molecular machines from different materials like proteins and DNA historically. We also discussed natural nanomachines like ATP synthase. In this discussion, we have covered much of what is turning out to be an exciting field in biotech. Learn about these things. Interesting to see how frontiers in biophysics are demonstrating capabilities which surpass all expectations, with no signs that we are near the limits. I would like to see if the turbines, when driven by hydrodynamic flows, are capable of, or could be designed, to store or convert that energy. It is clear that systems involving multiple turbines and providing voltages could be enough to produce smaller molecular factories. Creating nanopore substrates with the lithography step seems feasible for many labs and industries, so it remains to be seen how adoption of technologies like this will pan out in the decades and centuries to come. Until then, hope you have a good time and hope you learned something. Please check out below for sources and more information and a reference to the paper with this script. Goodbye.